on YouTube, I asked them to wait a little bit. We will be also giving you instructions there. We will start in about a minute. Chetan, when the YouTube link is live,
it's live, sir.
It's live. Please share the link in the chat as well.
I'll do that.
Okay. Okay. So we are live on YouTube and we have a good number of participants. Everyone who has all the students who have been attending, please welcome to the Science Setu series. We will be beginning with it soon. But first, just a few housekeeping instructions. For all of you, just we want to say that audio settings and all have been and chat box has been disabled, but the Q&A button is enabled. So you can always, if you are joining from a mobile or a desktop app, please please put your questions or any other problems that you have in the Q&A button. We will be monitoring that and we will address those questions. While the talk is going on, please put your questions in the Q&A box as well. So towards the end of the talk, we will be taking those questions and Dr. Bhavna will answer as many of those as possible. For all of you who are watching this talk on YouTube, we will be monitoring the live stream. Kindly put your questions in the comments on the YouTube video in the comment section. We will be copy pasting them and putting them into the Zoom call where Dr. Bhavna is there, and she will also answer the questions from YouTube as well. So we will keep you involved as much as possible. And with that, I will hand it over to Professor Apurva Sareen, Director of inStem.
Hi, good morning, everybody, and very warm welcome back. We had a longish break because of the way the weekends pan out. As Chandrakant has already told us, we really look forward to your questions, which we take at the end of the session, as we have been doing all along. I will be moderating them so that we cover as many of them as possible. Another point that I want to emphasize here is that we really look forward to your feedback. And I want to thank those who have started submitting artwork inspired by the talks that you have been hearing. So please keep those coming. We will find a way of putting it all together and having an interactive session. It's very motivating for us to hear back from you. Also send us feedback at the email ID that Chandrakant will post, I think, soon. It's very nice for us to hear from you when you tell us that you enjoy what you're hearing, and this is very inspiring and keeps us going. But more importantly, please let us know if there are areas that you'd like us to cover. As for those of you who've been with us from the very beginning, I said, we know that this is going to be, fortunately for us, a long interaction. It's going to at least go on for this whole year. So we have plenty of flexibility to take on topics that you feel you would like to hear about within the broad area of science, and particularly areas and questions related to biology. So feel free to send us that feedback. I can't say that we'll be able to have a talk for every idea that comes in, but we'll certainly do our very best. And then, of course, I think we just somewhat gloomy, but I think an important reminder that, much of the country is still fighting, and all of us are continuing to be impacted by the SARS-CoV-2 infection and the pandemic itself. I'm sure there's no one who's untouched, but I hope by and large everyone has stayed safe. A reminder that please double mask. Masking is really the one way we know that we can not only keep ourselves safe, but also our communities. And as I think budding scientists, people with an interest in science, people with respect for science, I think we should take this practice. We should also propagate it. So please make every effort to double mask and also to maintain distance and, of course, hand washing, all of which blocks and prevents the spread of infection. So with that, it is my great pleasure to welcome our speaker of this morning. This is Dr. Bhavna Muralidharan. She is a member of the faculty at inStem, and she joined us just a few years ago. So she is someone who I think still remembers all the trials and tribulations of being a postdoc, maybe not so much of being a graduate student and probably less so of being an undergrad, which is our community today. But I think you will hear a talk which is about a very complex structure, the brain. Bhavna's laboratory has been interested and is developing a program that is trying to understand events that occur during development to see how they shape the functioning of the adult brain. And we're going to get a glimpse of this in her talk today, which is titled Exploring the Brain, Its Birth and Function. And with that, I'll hand it over to Bhavna. Over to you.
Thank you, Purva. Thank you for the introduction, and good morning, everyone. And I'm so happy and excited to be talking to you today in a virtual mode. And thanks to Purva and the Comms Office for enabling this. So I'll begin by sharing my screen. Just here. So can everyone see my screen?
Yes, all good, Bhavna. Please go ahead.
Thank you. Yeah. The smell of this rose here, or even, the colorful butterfly, all of this perception and the experience is possible because of this 1.5 kilograms of mass which sits on the top of your head. This is the brain, and this not only does the bodily functions, but it also is a seat for your language, learning, memory, and consciousness. So today in my talk, I'm going to tell you about the structure function of this brain. I'm sure all of you would be up for some optical tricks to understand the function of the brain. So let's start with some optical illusions just so that we warm up. You don't have to, necessarily type in or anything, but just keep following whatever I say to understand what this illusion is. If all of you are looking at these two pictures here, and I ask you to focus where my pointer is currently, which is the right side of the screen, and also focus on this image here, which is on the left side of the screen, and ask you a question. Of the two images, which do you think is a lighter shade of blue? I'm sure many of you would agree with me when I say that the one where my pointer is the lighter shade of blue. I'll just give you a few seconds to process this if all of you are looking at this illusion along with me on your screens. So the one where my pointer is, if you all agree that this is indeed a lighter shade of blue, I'm going to tell you that it is not the case. If you connect the two images, you would see that they're actually the same shade of blue. So what is happening here? So when your brain looks at these two images, this and this here, it just not looks at the, the light coming from these images, but it also looks at the background from which the image is coming. And in this case, the background is lighter, the contrast is more. In this case, the background is darker, the contrast is less. So this appears lighter in shade than this. So the brain just not take images from here, but it looks at the entire picture and looks at the background as well. Okay. So this is a blind spot illusion. And today, if you're not able to do this with me, it's okay. Just Google and say blind spot illusion, and you'll be able to sort of do it. But if some of you, if you're sitting a feet away from your laptop, just as I am, all you have to do for this illusion is close your right eye and focus with your left eye on that plus sign there and move your head a little. And I think very soon you'll hit a spot where this dark black circle goes away, right? And I'll again give you a few seconds if some of you are doing it with me. Just look at me the way I'm doing it. And so there'll be a spot at which this black circle there goes away. And instead of that, what you see is the background of this light pale cream color. right? So the reason why the black circle goes away is because it's anatomy, pure and simple, because it hits the blind spot. The blind spot is a region in the retina where there are no nerves. But what is interesting is when the blind spot when the circle hit the blind spot, the dark circle went away. But what the brain did was, instead of showing you a gaping hole there, it actually showed you the background. It used the background around it and just filled up the blank. So basically, the brain did not want you to have the perception that the world has a gaping hole in it because of your blind spot. And just one more illusion, and sort of this will again help us in understanding how the brain functions. So look at these two images. And if I asked you, which one would you like to name as Booba and which one you would like to call the structure of the image here as Kiki, again, I'm sure many of you will agree, just like many people did when Villeneuve Ramachandran and Edward Hubble did this experiment several years ago. Many of you will call this Booba, where my cursor is right now, and call this one as Kiki, where the one which has sharp corners. So what your brain has done is taken the the round curves in this image, and also taken from the auditory system the way Booba is spelled, right? You're rounding off your vowels as opposed to Kiki, which is more rugged, like the very corners as you've seen in this image. So this is like a cross-modal extraction which your brain has done, and immediately assign the word Booba to this and the word Kiki to this. So how does the brain does this? So let's take one very simple example of the vision which we just saw and see how the brain extracts information. So what I'm trying to tell you is the vision, or the vision will happen in the eye. The eye may see, but the perception and the experience happens in the brain. So the visual perception begins in this wonderful system called the eye. And like if this light from this pencil actually hits your lenses, which is there in the eye, and an image is formed in the retina. So the retina converts this light signal light into an electrical signal, and this signal then goes to the brain, where what happens is this. So the image which is formed at the retina is converted into an electrical signal, and from there it goes into a relay center in the brain called thalamus. Now, why do I call this as a relay system, a relay center, is because not only it gets in information from the outside world, like the eye or the touch or the ear, it also gets information from the brain, from the higher order centers in the brain, and passes it on to your sensory apparatus. So that's the function of this relay system, thalamus. So the signal from the retina goes into this thalamus, and then it goes to a primary visual area called B1. And from here, the information is split. So if you're looking at a colorful object, then the information is split into two streams. One is called the watch stream, the other is called the where stream. The watch stream will extract information on color and form and send it to a temporal pathway. And the where stream will extract information like distance or movement, and then send it to a different area in the brain called the parietal pathway. And there in the higher centers of the brain, the information will be processed, decisions will be made, they will be assembled together, and a decision will be made. Much like this hypothetical model, and let me tell you, this is a very simplistic model, it's not as simple as that, but just for our understanding, what really is happening is that if this is the visual system here, and this is a red color object, then the first feature map which is made is actually split, where information like color is processed separately, orientation, size, and distance. And this then goes to the higher processing center, where each of this is made sense. For instance, the brain decides whether to take, I should just not focus on the light which is coming from the blue part of the image, but I should also look at the sort of contrast or the background. And that's why you see one shade to be lighter than the other. Or for instance, when you see a blind spot, the brain understands that the world can't have a gaping hole. So it takes image or information from the background and makes it uniform or smooth. Or for that matter, it does cross-modal extraction, where it says, okay, this audio is very rounded and this image is rounded. So this is Booba and that is Kiki. So this is what is sort of happening. And because all of this happens in such fractions of seconds, you never realize that the brain is constantly doing this. So what enables the brain in doing this? In other words, how does this function come about? So for the brain to do this function, its structure should be built in such a way that it has all of these different kinds of neurons. The billions, it has about, the billions of neurons which the brain has. Many of them have to code, have to do the color processing, the orientation, the size, and the distance. So it has to have, the structure should have all these different kinds of neurons. The brain not only has neurons, but it has support cells called the glia. So the brain has to have, the structure should also have this glia. And all of this, and one very important feature is all of this have to be connected to be talking to each other. So the brain should have connections as well. So that is where the emerging function comes in a nervous system. It starts with molecules. It's very molecular. Molecules molecular processes enable the formation of cells. And in this case, neurons and glia, which are the two cell types in the brain. The neurons are the firing unit, which fire up and pass on the signal from one neuron to the other. The glia are the support cells, which nourish and support the neurons in this process. And then these neurons and glia have to form connections or functional circuits, which can talk to each other and enable the brain to do its myriad of functions, which it does. And finally, that leads to perception and behavior. And this structure is built actually, the billions and trillions of cells in the brain are actually built up from a small pool of stem cells during development. And today in my talk, I'm going to tell you further how this structure is built. And so the first step starts with, so during development, the embryo and particularly the neural tube consists of stem cells, which are called radial glia. And these have the capacity to undergo two kinds of divisions. One kind of division is called symmetric division. As the word says, the cell divides symmetrically into two daughter cells, and both of them become stem cells. Now, this is a very important feature because to sustain the entire period of development, in case of humans, which is about 9 months, you need enough number of stem cells which can divide and give rise to all the kinds of neurons and glia which you see in the brain. So these stem cells undergo symmetric division, they proliferate, they generate more of their number, they make enough so that the entire development is sustained. Then they switch to a mode of division called asymmetric division, where these stem cells give rise to two different kinds. Asymmetric means you give rise to two kinds of cells. One is a daughter cell. One daughter cell is actually post-mitotic or a differentiated cell. In this case, it's either a neuron or a glia. And the other, again, it gives rise to a stem cell. So this is how the process of neurogenesis starts in the brain. So it starts with stem cells dividing, then followed by formation of neurons or glia. Then these neurons and glia, you can see here that this is a neuron being generated from this progenitor here. Then these neurons and glia have to have their axons elongated so that they can elongate their axons and then form connections. And that is axonal outgrowth and connection, finally leading to a functional network, just as you see here in this colorfully in this brain, which is showing the various different networks or connectivity or circuits which are formed between different parts of the brain to let the brain do its function. So up until now, and today in my talk, I'll tell you about my own work where I've studied how the stem cell gives rise to this, two different class of neurons. And the neurons which I'll be talking to you about, I spoke to you in a few slides before. Some of them connect via the relay center called the thalamus. Right. So till now, I've been talking to you about this brain. All of you have seen these images on the internet and everywhere. But this brain is not that accessible. So most of our studies regarding how the brain functions or how the higher order center function actually comes from this brain here, which is the mouse brain. And immediately looking at this, you will say that the two look different. And one stark feature is that this brain here consists of folds, but this brain here is smooth. And I'll come to that in a bit. But right now, let me tell you some of the study which I've done using this structure here. And many of these features are conserved. So this system here, indeed allows you to study such conserved features and molecular mechanisms. And the first, I'm going to tell you about how I've used the mouse brain to study some features of how this brain is built, or how the neurons are being generated, and then tell you some of the features which are altered or, between the mouse and the human, and how my lab is going to use another model system, which I'll be coming to you in some time, to study altered features and mechanisms in human in vitro disease models. So coming to understanding conserved mechanisms in mouse, this is how a mouse developing brain looks like. So this is at, say, an age called embryonic day 12.5. So in mice, the mouse gestation is about 18, 19 days, which means if day 0 is the day when the sperm fuses with the egg and generates the zygote, then on the 18th or the 19th day, the postnatal pups are being generated. So this is the developing brain at an age called embryonic day 12.5, which is 12 days post-fertilization. So if you cut your brain just like this, face-wise, just like this, you would see a brain which is hollow in between because it consists of a nourishing fluid called the cerebrospinal fluid. And then what you see is this region here, which is yellow and red. So this region here in yellow is all the proliferating cells. And as you can see that at this age 12.5, which is about 6 or 7 days from gestation, from delivery or birth of the pups, the epithelium or the primordium, which we call, or the brain, developing brain, is still mostly consists of stem cells. And there is just a very small, post-mitotic cells or the neurons which are being born. So the question is, how this stem cell here, population, or the cells which are full of stem cells here, then go on to generate this neocortex. And I just described to you that the brain has to do a complex function of, extraction, cross-modal extraction, of, making a sense of your visual world, of your auditory cues and all of that. And that is possible because of this 6-layered structure here called the neocortex. So the neocortex has 6 layers of neurons, and each layer has a different function. So how does this then simple looking stem cell population become this 6-layered structure? So what happens is that this is a regulation which happens of timing that at each and every day in development of the brain, at each and every day, this step of generating a new class of neurons is regulated. So this is at embryonic day 10.5, 11.5, when the epithelium is mostly with stem cells. So these are the stem cells. And the very first day, which is about E11.5, these stem cells, as I told you, they undergo asymmetric division. They give rise to a neuron. That neuron goes out and sits on top. And the neuron which is being generated at this age is called a layer 6 neuron. And in an adult brain, it would look like this, which is here marked with the transcription factor TBR1. So the very next day, what happens is the very same stem cells, they switch, and then they, instead of producing layer 6 neurons, they switch and they now start producing layer 5 neurons, which will be generated here where my cursor is, migrate past this layer 6 neurons and go and sit on top. And that is the layer 5 neurons. So this happens the very next day, which is the embryonic day 12.5. And here marked with the transcription factor FISF2. So this is TBR1 layer 6, this is layer 5, which is just on top of that. This is then followed by generation. The very next day, the switch happens and you produce something called layer 4 neurons, which again is produced here by these stem cells. They migrate 6, 5, and they go and sit on top. This happens at 13.5. And so on and so forth, till you generate all the 6 layers in the cortex. And here you can see that this is 6, 5, 4, and these are the upper layers 2 and 3, 4, 3, and 2. So, sort of temporally or regulate the timing of generation of these neurons. And this is very, very important. So when I studied, wanted to understand how this regulation is happening, we looked at a transcription factor called LHX2, and we looked at its expression in the cortex. So this is this embryonic day 12.5 brain. And as you can see and recollect that I told you that at this age, most of these cells are stem cells. And you can see that LHX2 marks all of the stem cells in this at this age. But very soon, 3 days later, you can see here the stem cell pool is, of course, you can appreciate that the brain itself is growing, right? And the stem cell pool is now reduced here. But what is also growing here is this post-mitotic neuronal cells. And you can see here that LHX2 is expressed still in these proliferating stem cells, but it's absent from the neurons. And specifically, it's absent from those neurons which were produced by this age, which are layer 6 and layer 5. And it postnatally, this is a postnatal brain, it comes up in the upper layers, which are layer 4, 3, and 2, but it's still absent from layer 6 and 5. So this led us to ask the question, does LHX2 have any role in regulating this timing of layer 6 and layer 5 neuron production? So to understand this, what we did was we took a mutant of LHX2. So essentially a mutant means you are either knocking out or knocking down the expression of the particular gene, in this case LHX2. And we used a very specific way in doing this so that the removal of LHX2 happens only in the area of the brain, which I call as the cerebral cortex, which is the seat of all higher order functions. And also the timing is very important. So we time it in such a way that the removal of LHX2 only happens at, embryonic day 11.5, so that any other functions of LHX2, which is important for its other functions, is not disrupted. So when we did this in such a LHX2 mutant, what we saw, so again, this is the control brain, and you will now immediately recognize that here TBR1 is layer 6 and FESF2 is layer 5, and two other markers for layer 5 are ER81 and IT2. And in the LHX2 mutant, what we saw was a decrease in layer 6 neurons, which is seen here in reduction in the expression of TBR1, followed by an expansion of layer 5 neurons, as you can see here from the expression of FESF2, ER81, and IT2. And this is just to show you again that it's indeed the numbers are altered. So here this is TBR1, you can see it's reduced, and this is CTIP2, another marker for layer 5 neurons, and you can see it is increased. And this is just to show the quantification that indeed CTIP2 is increased and TBR1 is decreased in this mutant. Right. So now coming to, what are these neural subtypes and in these layers, in these different layers, and what is their function? And this is where I think it'll help you connect with what I told you in the very first place about the visual system and how signal goes from the relay center and so on. So the layer 6 neurons, which is marked with TBR2, TBR1, is actually the corticothalamic neurons. Now, if you remember, the signal from the retina, the light signal which got converted into an electric signal, that signal from the retina came to this relay center, right? And from the relay center, it went to the higher order centers, which is the cerebral cortex in the brain. Now, once a decision is made, it comes back from the cortex to the thalamus. So whatever carries from the thalamus to the cortex, those are called thalamocortical neurons. But information which comes from the cortex, once a decision is made, it comes to the same relay center called thalamus. And those are the corticothalamic neurons. And those actually come from the layer 6. So you can see here that there are 3 layers here. The one which is in green is layer 6. The one which is in red is your layer 5. And here in blue, for simplicity, is the upper layer neurons. So the 6 is what projects from the cortex to the thalamus. And these are called the corticothalamic neurons. So where does 5 project? The 5, which is actually consists of 2 different classes. One is FESF2, CTIP2, which I showed to you. These are subcerebral neurons, or one class of them are ones which project to the spinal cord. So if you can see here in my thing, you can see the red neurons, which are actually going to the spinal cord. And these are very important. These are called the corticospinal motor neurons, which are important for your movement, which allow for all your movement to happen in your body. The other class of neurons are the SADB2 positive neurons, which are marked by a transcription factor SADB2. And a class of these are the callosals. And these actually are the ones which help connect one half of your brain to the other half. As you can see, this blue is going from the right side to the left side of the brain. And so these are the callosal neurons. So these are the different kinds of neurons which are happening. And you can appreciate here that the brain, for its function, actually needs these different classes which are different anatomies, where different molecular structures, which are different connections. And because of these different connections, it's able to do its function. So, so these are the different class of neurons. And if you now remember that in the LHX2 mutant I showed to you, there was an increase in one class of neurons, which is the FESF2 CTIP2, and a decrease in the TBR1. So let's now look at how this switch is being regulated. So when we were doing this study, a bit was already known about how this molecular regulation happens. That is the switch from the layer 6 to the layer 5 production. So for the layer 6 production, two transcription factors, TBR1 and SOX5, they're absolutely important for the production. They do so by actually inhibiting FESF2, which is important for making layer 5 neurons. So they inhibit FESF2, and that's how you actually, end up, and they're activated, so they help in the production of layer 6 neurons. But when the switch has to happen, two other transcription factors, SOX4 and SOX11, quite literally knock off SOX5's inhibition, and they activate FESF2. Once FESF2 is activated, it in turn goes back and represses TBR1. So there is like a feedback, represses. So now TBR1 levels are reduced, SOX5 is knocked off from the FESF2, the repression. SOX4 and SOX11 activate FESF2, FESF2 becomes active, it activates CTIP2, and that's how the switch happens from layer 6 production to layer 5 production. And separately, in some neurons, which I'm not going into details now, they get SADB2 expression, and once a neuron gets SADB2 expression, it then represses CTIP2. So the two different classes in the same layer, namely the callosals and the subcerebrals, they get segregated out. So with this in mind, we wanted to understand what is LHX2, the transcription factor, doing, or what's happening in that mutant. So we did a technique called ChIP-seq. Not going too much into details, this technique, powerful technique, allows you to understand protein-DNA interactions. So if you have a transcription factor, which is a protein, you can see to which region in the DNA it is binding. So we precisely did that. And what we found was LHX2 was binding to the DNA or the regulatory elements of the gene FESF2 and of the gene SOX11. And so this here again is just showing that indeed it's LHX2 is bound at FESF2 and SOX11 locus as opposed to a region which does not have an LHX2 binding site. So now I showed you a mutant in which we looked at postnatal or the adult brain and seen that the layer 5 was expanded at the expense of layer 6. Now, the moment you remove LHX2 within a day, what is happening to the expression of these two transcription factors, FESF2 and SOX11? And what we saw here was the moment you remove LHX2, there is an upregulation of the expression of FESF2 in the mutant cortex. You can see here that, what is so less has actually become upregulated. This is continued even at embryonality 13.5, and same is true for SOX11 as well, which you can see here, right? So within a day, you're seeing an upregulation of FESF2 and SOX11, and this is then sustained, and that's why you see an increase in the production of layer 5 neurons at the expense of layer 6 neurons. We also wanted to understand sort of some, because this is a transcription factor, it has to interact with other factors or chromatin factors which sort of regulate the chromatin, or because the way the DNA functions is that there are regions in the brain, regions in the chromatin or the DNA which is closed, that has to open so that you can allow the expression of genes. So we wanted to understand that LHX2 does it interact with some of these modifiers which help in closing or opening the chromatin. And indeed, we saw that LHX2 was interacting with 3 such chromatin modifiers, LSD1, RBBP4, and HDAC2. And not going into too much details, we also saw that by the same technique called ChIP, chromatin immunoprecipitation, we saw that even LSD1, RBBP4, and HDAC2 are bound at the regions where LHX2 is bound. So this here shows that LSD1 is bound along with, at the same region where LHX2 is bound on the FESF2 locus, on the SOX11 locus. RBBP4 was only at the SOX11 locus, but HDAC2 was again found at the LHX2 binding region. So which means that perhaps LHX2 is interacting with these modifiers. The modifiers then enable the opening and closing of the chromatin. And then that leads to, the repression of FESF2 and SOX11, just at the time when the layer 6 production is happening in the cortex. And then we also looked at epigenetic marks. So once the chromosomes, the chromatin is open or closed, that is also regulated by some of these histones, which enable this process of opening and closing on chromatin. And active marks means the histones, if they have active marks, then the gene will be expressed. If they have repressive marks, the gene will not be expressed. And we saw that the moment you remove LHX2, the active marks are actually going up in the mutant, and particularly at the region where FESF2 is being expressed. So this suggests that, the regulation happening by LHX2 is via interacting with chromatin modifiers, which then lead to a change in the epigenetic marks, and that regulates the expression of the gene. I mean, this is just simply to show you that now, what if you overexpress LHX2? Can you now suppress the CTIP2 or the FESF2 layer 5 specification? And indeed, it's true. And here we've done a technique called in utero electroporation. So we go into the brain at embryo day 12.5. We do a perturbation, we stimulate zap electrodes, put in DNA, we overexpress, and then we close up the mother. This is like a surgery which is performed. And postnatally, we look at the brains. And I just wanted to show you how a mouse brain looks like. And these are some of the neurons which we had electroporated. You can see the GFP. You can look at the neurons here. And this is just plain GFP, but this is overexpression of LHX2. So what happens is, at embryo day 12.5, if you do a control electroporation, you see about 57% of them are CTIP2 neurons, because that's what is being produced, layer 5. But the moment you overexpress LHX2, the number gets reduced to about 30%. And then those neurons, which are now not CTIP2 positive, actually become SADB2 positive. So there is a switch the moment you overexpress LHX2 happening from a neuron which ideally should have been a CTIP2 positive, has now become a SADB2 positive neuron. And just to summarize this work which I showed to you, which I published during my postdoc, is that in the presence of LHX2, these chromatin modifiers are able to access the locus of FESF2 and SOX11. And they keep the active marks at a reduced condition, and they keep FESF2 SOX11 repressed. But the moment you remove LHX2, these chromatin modifiers, which are actually they remove active marks, they're now not able to access either the enhancer or the promoter region of this FESF2 and SOX11. The active marks then go up. There is an upregulation of FESF2 SOX11, which actually leads to increased production of layer 5 of subcerebral projection neurons. So this is the switch, and here we've shown that this transcription factor represses FESF2 and SOX11. So I told you about some of the conserved features which I studied from the mouse brain. But what I'm going to now tell you is how this, we would now, my lab at inStem would like to study some of the altered features or mechanisms from human systems. And this is where now in my talk, I'll be telling you about how my lab is now focused into doing discovery biology of neuropsychiatric syndromes. A study in 2017 published in Lancet Psychiatry put the number of mental illness patients in India to be about 197 million. That is about 1 in every 7 Indian is suffering from a mental illness. So this is a huge socioeconomic burden, and there are no targeted therapies for these disorders. So it is very important that we study such disorders to understand what are the pathological mechanisms or the disease mechanisms in such disorders. And a large number of these mental disorders are actually neurodevelopmental disorders. That is the disorder, and if you look here, I have highlighted some of them: the intellectual disability, schizophrenia, bipolar, and autism spectrum disorder. So what is a neurodevelopmental disorder? A neurodevelopmental disorder is when the pathological mechanism leads to a disruption in brain development. So this happens during brain development because of genetic risks or environmental factors, which then result in a sort of affected, function in the adult or even in teenagers. So these are neurodevelopmental disorders. And my lab is interested in understanding the mechanisms of pathological mechanisms or disease mechanisms of a disorder called schizophrenia, which is a disorder of aberrant perceptions, thought, and social connectivity. And many family studies show that it's heritable. So about 70 to 80% of this is heritable, that is, it's herited or it is genetic. And as I mentioned, it's neurodevelopmental, which means the disruption happens during development. Some of the steps which I just showed to you as in the mouse model system. And there are many studies from Caucasian populations and very recently from African populations as well. But given the high degree of homozygosity in the Indian genome, I think it is important to study this from the Indian population as well. And now coming to back to the mouse, and I showed you some conserved mechanisms in the mouse. Now when we look at the human developing brain, and this is a mouse developing brain at embryonic day 13.5, some of you will just immediately recognize because I just showed to you that how it consists mostly of stem cells here, which is in red and green. But the same age which is gestational week 14 for humans, you would see that, yes, there are something which is similar, but there is one region which is called the outer SVZ, or an additional layer of proliferating progenitors, which is entirely not, which is sort of not present in the mouse or very little in the mouse. And this region called the outer SVZ has been implicated for the neocortical expansion, which is seen in humans. So the cortex or the neocortex, or the region which does all the higher order processing, is so expanded in the humans that it was not able to contain it within the skull. And to do so, actually, the cortex got folded. And that's why you see this gyra in sulci, or the folded cortex. And this can be very easily seen in this cross section. You can see the cross section of the mouse is smooth. You see this is the neocortex here, but if you look at the cross section of the humans, you will see that it consists of a lot of folds, which is because of the expansion, and that expansion actually has been attributed to this zone of extra proliferating progenitors in the developing human brain. So there are clearly differences between the mouse and the human. So you need systems, or you need model systems where you can study such differences, particularly when you want to look at patient-specific mechanisms, because you want to also factor in the genetics of the disease. And that's where the system of iPSCs, or induced pluripotent stem cells, have become a very valuable technology, because the human brain or the fetal brain is not accessible that easily. You need an alternate system where we can understand some cellular molecular mechanisms just the same way I showed to you in the mouse model system. So what are these iPSCs? So one takes skin or blood cells from adult and then adds certain factors called Yamanaka factors, which are deprogramming factors, which will convert these skin or blood cells into pluripotent stem cells. These are similar, very same as the stem cells which are seen in a growing embryo. And these are pluripotent, so they can give rise to all the 3 germ layers in the body. Then you can use these stem cells and give them a cocktail of factors to specifically derive very specific tissue or tissue types or cell types in the body. Many people use it for deriving liver, lung, pancreas. My lab utilizes these iPSCs to sort of make mini sort of cerebral organoids, which are small tissue-like 3D structures which mimic human brain development in a dish. So we take these iPSCs and give them a cocktail, grow them in petri dishes to make these cerebral organoids. And these organoids are very, very valuable to study human brain development. And as I said, since this comes from an individual, it also carries the genetics. So which means if you want to understand the genetics of the disease from an individual and understand the disease pathology, this is a very valuable system to do that. And further, I think the future will be drug screening using these organoids to enable precision medicine in the future. So my lab takes these iPSCs, and these iPSCs actually come from a program called the Accelerator Program for study in brain disorders using stem cells. This is a multi-institutional program which is of which NCBS, inStem, and NIMHANS is a part of. And what this program is that it's a study to understand neuropsychiatric syndromes. What are some of the attributes of this program is that there exists a large cohort of clinically dense families which have major psychiatric disorders. And these families, there are at least greater than 2 first-degree relatives who have the disorder. And the program also has unrelated controls, so that there are controls and there are patients in the study. Further, these patients have their clinical symptoms are being described so that one can arrive at a precise clinical definition. And what interests researchers like me, developmental neurobiologists who want to understand mechanisms of neurodevelopmental disorder, is that iPSCs are being generated from these patients, and also their exomes have been sequenced to understand the risk or the genetic risk factors which could be behind these disorders, so that people like me are enabled to study the genetic and cellular basis of disease development. So my lab, as I said, uses these disorders to do human in vitro disease modeling. We have chosen one clinically dense family, which has schizophrenia. The exome sequencing from this family is available, so which means the putative risk variants probable, which could contribute to the disease, to the genetics. We know that to understand what could be, contributing to the disease. And finally, we will be performing cellular and molecular studies. And to match that, the clinical assessments are already in place to correlate that, whatever cellular and molecular mechanisms we'll understand, there'll be correlates in terms of the clinical symptoms which these patients show. So as I said, I'm interested in one such family, which is dense with schizophrenia. And you can see here that all these black ones are the ones with patients, and there is also this control here. And upon exome sequencing, a risk variant, a putative risk variant, was identified in a chromatin modifier, something which my lab is again interested in. So this is SED6, which is an histone methyltransferase. So it methylates the histones. And there was a risk variant which was identified, which was seen in all of the patients, but not in the control. So now with these iPSCs of these patients, which have this putative risk variant in SED6, we are asking the question that from these iPSCs, if we make them into organoids, and then if we generate, these organoids will generate all the different steps in development, as I told you before, going from proliferation to generation of neurons to axon growth and finally formation of synapses. We're asking the question that, what are some of the epigenetic or gene expression changes which can come from the risk variant in SED6? And then that leads to an altered cellular phenotype in the dish, so that then we can understand that how a risk variant may lead to an altered cellular neurodevelopmental phenotype, which is seen in a dish. So I started by telling you about the emerging function in a nervous system. And I told you that normal brain development, or for that matter, the emerging function goes like this. It goes from molecules to cell, it then goes to circuits, and then leads to behavior. What my lab wishes to understand is what happens in brain disorders, because there is a genetic change which has an impact on protein activity that impacts the cellular function of a neuron, or it has an impact on the neuronal function. That then leads to a dysfunctional circuitry, because the neurons, which are such a key neurons, or the neural cells, neurons or glia, which are a key part of the system, if they are affected, their cellular function is affected, then the circuitry which will be formed will be dysfunctional. Then that leads to affected behavior, and that is what comes off as clinical symptoms in patients. So this is just a schematic to show the same, how mutations or altered epigenetic mechanisms lead to altered neural development, lead to a dysfunctional brain. So that brings me to the end of my talk. And with this, I would like to thank my postdoc mentors, and these are all my current lab members. I would also like to thank all the funding agencies, and thank you all for your patient listening. I'm very happy to take questions now.
Thank you. Thank you very much, Bhavna. I think you took us on a really sort of detailed journey going from molecules to perhaps function. And in fact, some of the questions started coming up really early in the talk. So I'm just going to try and consolidate some of these so that you can explore ideas. You want to keep your any of your model slides on, or you know,
sure,
whatever's good for you?
Yeah, I don't mind. Yes,
I'm just fine with this. So we'll start with a question that refers to the early part of your talk. Now the question here is whether how does the brain? I mean, does the brain use different cues for each object that it sees? I think this goes back to the Boba and Kiki images that you
Okay.
showed us. The question is that, will the feature map be distinct for every object the brain ever encounters?
So I think I have to say that I'm not an expert.
Shape perception, I think, is the question here.
Yes, yes. So, I mean, let me just say this upfront that I'm not an expert on this. I'm not a cognitive neuroscientist. I'm a developmental neurobiologist. But yes, broadly the answer is yes, because the brain does look at, does do shape selection. How is it known? Is that if the orientation of a lighted object, if it were horizontal to vertical, the brain is able to see these differences. So yes, clearly it does look at the shape, whether it's horizontal or vertical, it does do that. And that I would say is purely from what I have read. Some experts may be able to give more detailed answers as to how it changes or whatever.
Okay, so there's another question from Akila Karani, who had asked the previous question as well. And I think some of it was touched upon in your talk. The question is about an artificial brain being produced from a stem cell. So I think your comment, you may want to just summarize how much we can learn from organoids or not, maybe.
Okay, yeah. So, I mean, organoid is not a brain. So let me just say that upfront. So what we are actually producing in the dish is some regions of the brain which we are interested in studying. My lab in particular is interested in studying one region of the brain, which is the cerebral cortex. Particularly, that is the region where your higher order functions and 6 layers, which I described to you, happen. What we are trying to do is here trying to, give these stem cells. So of course there is 2D also, where we are using the same stem cell, we are producing the different neurons. But since brain is a 3D structure, in a 3D structure, the connectivity is much better between these neurons as opposed to in a 2D culture. So right now, I would say the field is in a space where we are able to produce these stem cells. We are able to produce all the 6 layers of the neurons. We're able to produce the different cell types, and we're able to ask very fundamental questions on how these talk to each other, like in terms of synapse formation and all of that. But long range connections and all, it's still very distant. I would say mouse is still a model system to do long range connectivity.
Okay, and here's another, there are lots of questions, Bhavna, so we'll go through them. So here's another one from Anchal St. John's in Kerala, Amal Sundar, asking the question as what percentage of a human brain is actually working? I don't quite get the context, but I suppose in a particular task, the question is how much of a brain would be involved in a task? Do we know that?
So I think her question is coming from the fact there were some studies going around, not studies, there were some articles in, out there in social media or saying that you're using only 1 to 2% of the brain. I don't think that is true. So at different time points, we are using different regions of the brain. So, I mean, I don't think there is any number as to say how much of your brain is actually involved, because there are things like when you're focused or when you have attention, you're very focused, right? So you're using one part of the brain as opposed to doing different, when you're just observing or something. So there's no clear number as to what percentage of brain you're using.
Okay, so now we'll get on to some slightly more questions related to the latter part of the work and the more technical elements. This is Sri Vaishnavi Ranganathan asking that can we use this approach, and I think that is the approach that you described in the lab at inStem, to study mental health disorders such as depression and eating disorders.
Right. So I think I'll have to say this, that, this system is very, very useful to study, to capture early development, neurodevelopment, for understanding some of these disorders which come up late, which are adult psychiatric disorders. Right now, we're able to keep these organoids in the dish for about 1 to 2 years. And I don't know whether that is enough to mature these cells so that they are exactly like an adult brain. So I think we still have a long way to go there, but I think these disorders are useful to study some of the early neural developmental phenotypes which we see. So neurodevelopmental disorders, yes. Depression and all, I think we need to moderate or change our systems so that we can mature the brain enough for it to go there.
Okay, then a question from Srinivas Prabhu says that asking about the timing of developmental decisions and how these are impacted by events that occur earlier. So could expression on previous days have an effect on functioning after day 11.5? Do we have a mechanism to address this? So I think the question is that how do you study early events in development in functioning of the brain, if I've understood the question correctly?
Okay, so I think I'll slightly, perhaps with slightly changing it, I'll be able to answer this. So some of these transcription factors or even modifiers are expressed throughout in development, and their function seems to come and go at specific developmental time points. So which means that, everything has a role at a particular time point. So to chalk this out, we use different ways to express. As I said, we time it, we use both spatio-temporal, lines or we call them driver lines, so that, we can specifically remove, say, expression of a transcription factor in a zone spatially, only in the progenitors or only in the neurons. That's one way to do it. We also time it. So we have Cree driver lines, which can time and where you can enable, probably at 8.5 and stop it at 10.5 to understand, whether these have roles much before than 11.5 as well. And in fact, for LHX2, we have done some of this, and we know that it has an earlier function as well.
Okay, so we encourage the person to ask again if the question that you originally posed has not been addressed, because we have a few more questions to go, so we will be here for more. Now, this is a question about the temporal regulation of, you said, of LHX2 in your talk. So the question being asked is that its influence on PETSEF and SOX transcription factors has also been shown to be spatial. Could you comment on that?
Sorry, what was the question again?
So it says, my question is that you talked about LHX2 as temporal regulation, but its influence on SOX and the
PETSEF2, yes.
PETSEF2, is it? Okay. Transcription factors shows that it also regulates them spatially.
Yes, that's a very interesting and a very nice observation, I would say. Yes, it does. But right now here we've looked at the role of LHX2 only in the progenitors. So the Cree line which we used, we very specifically removed its function from the progenitors. And there it seems to be repressing both PETSEF2 and SOX11 to ensure that, and these both, they actually come up in neurons. They don't come up in progenitors. So basically LHX2 is that mechanism or that transcription factor which makes sure that these don't come up precociously in the progenitors at the time of layer 6 production. So indeed, LHX2 is doing this spatially. And to understand whether it does this post-mitotically as well, we'll have to use a different Cree line to do that and understand.
Bhavna, a question about the blind spot illusion, which we should also encourage the person to actually go and check it out. But the answer, the question here is, if we cover our left eye instead, is this something that is related to handedness? So do you get the blind spot disappearance with both eyes? Or?
So I don't know the answer, but I'll tell you what I did. I covered my right eye and I used the left eye to see it. But if I cover my left eye, I don't see the illusion. So you'll have to swap it. So which means the blind spot is it's because of our vision is binocular, right? So the blind spot for each one of them is at slightly a different position. Right. And that then gets overlapped because of the binocular.
The question is it related to handedness? I don't know if you want to comment on that.
No, I don't know.
Okay. All right, then a question from Jerry Jackson, who says that they enjoyed your talk. It was simple and informative. The question here is that could you elaborate on the applications of organoid development with IPS, using iPSC? And the application that they want to comment on is drug screening. I think you just alluded to that briefly as you were speaking. Yeah.
Yes.
So do you have a comment on that?
Yes. So I must say that, the beginnings are there of using organoids for, for drug screening. And a few reasons why organoids are becoming a model of choice is that for many of these disorders, particularly for Alzheimer's, which is a neurodegenerative disorder, mouse model systems are not recapitulating aspects of amyloid or plaque generation, which the human systems are shown. So which means that even for generation of plaques, you need to have a model system which is human. So these changes which are there between the mouse and the human, which is actually rendering many of these drugs which are being found, which are being tested in mice, they're failing at many of these clinical trials. So people are now moving towards a system which is more human, at least the few basic steps of, perhaps making the neurons and all will be, they want to study in a human specific system. So that's where the use of organoids is there. And coming very specific to drug screening, people are now trying to incorporate for drug screening, they're trying to do organ-on-chip sort of models where there are organoids they're also trying to use microfluidics to mimic the blood vessels and all of that. So that will sort of tell you, but it'll be very useful for drug screening because you can understand drug metabolism and all of that in a human system. So that's where I think the field is going, and I think that's a future application, and people have already started those steps.
Bhavna, there's a specific question on schizophrenia. It says that you talked about the many factors that contribute to neural developmental defects. Question being asked is there a major epigenetic factor known that is causal or related closely with schizophrenia?
So yes, the answer is yes. Some of these chromatin modifiers have been implicated to be perhaps causative of schizophrenia. There is, this one, some transcription factors as well. So so schizophrenia is typically seen as a disorder of two things. One is very early development, the progenitor proliferation and the neuron generation. It is also a disease of the synapses. So there are these two things which are highly being often implicated as, mechanisms disrupted in schizophrenia. So there are either the synapse molecules or there are molecules of chromatin or regulation and all of that, which often come up as risk factors.
So while on these factors, Bhavna, there's a question asking whether these factors that you referred to are also implicated in neural tube defects.
Neural tube defects, okay.
That's CLIP2 and FESF2. Do they also correct?
They want to know whether FESF2 and CYP2, no. So they are not been implicated in neural tube defects, no.
Okay. Now, question on, which I think you may have to step back and clarify. The question says, can brain lesions which occur because of prolonged mental illness, I don't know, you may want to comment on that as well. Can these be corrected by surgery? First of all, are there brain lesions associated with mental illness?
So brain lesions are so in epilepsies, there are brain lesions, yes. I'm not aware of particularly schizophrenia or anywhere where there are brain lesions. So there are no structural abnormalities seen in the brains. If you were to do an MRI, there are no structural abnormalities, but epilepsies, of course, there are brain lesions, cortical lesions very specifically. Now coming to whether how to, I mean, is the question like a therapy? Like, can you?
Yeah, I think it is so, but we do understand, I think we have to take on board your comment that mental illness itself does not, the mental brain disorders that we're talking about and your lab focusing on, are not associated with lesions. In the brain. I should also inform our audience that one of our talks a little bit later in the series, will be by one of the clinicians who's part of the study that Bhavna referred to. And so, there is a clinician who's trained at LIMHANS, which many of you will know is one of the key centers for, mental health, both the study of and also the clinical components. So I'm sure that there would be questions that that could be directed and brought up again, from what you've learned from today. Now we have, I think, a very general question. I also note that there are questions on consciousness, which I will not take up here because I think they're way beyond the scope of what we are discussing. And I'm sure they're great for another discussion, another forum, another space. Bhavna, just a specific question about whether intelligence in any way correlates with the folds in the brain. And I don't, you feel free to see if you want to address it.
No, I don't think there's any study, scientific study in terms of done on many people to connect folds to intelligence.
Okay, we'll leave it at that. The question now here is about a specific question saying that, is it refers to all the interactions that you talked about, the molecular interactions. Okay? And the question again now is that is it possible to mimic all these interactions involving also different cell types using 3D organoids from iPSCs?
So I think the answer is no. Right now, it's not possible to mimic all the interactions. As I said, we form very specific regions of the brain. And for instance, like the brain has neurons and glia, but it also has, two kinds of neurons, the excitatory and the inhibitory neurons. And in fact, the inhibitory neurons come from a different source in the brain. So today, whatever I told you was all the excitatory neurons. And when we make organoids, we use a very specific region. So the region which I use is only the region which will go on to produce the excitatory neurons. Now, if I want to have the inhibitory component, I need to generate another organoid which will generate the inhibitory component. So which means, and the brain the interaction consists of both the excitatory, inhibitory, the glia, the microglia, and everything. And not all of it is possible right now to produce in one place, in one dish. So the answer to that is right now it is no, because we produce very specific structures with very specific cell types.
Okay, there is a question here about the gut-brain axis. I think this is a question about the microbiome impacting brain function, and the question being, how do you study this dependency in a laboratory?
I think right now it is mouse studies. You can study the gut microbiome interaction, and people have done this for autism and Parkinson's diseases. And that's the only way, because the mouse is a whole model system, which is like a live animal where you can look at these interactions.
Okay, so I think we have a couple of questions which are very clinical and oriented towards therapy and treatment. So I think we'll not address those, because it's also important that there be no information which is incorrect put out in a forum like this, because I know we have a really diverse audience. So with apologies to everyone who's asking questions about treatment, this is not an area that I think we should be commenting on lightly. And I'm going to leave that till when we have somebody who can perhaps speak in this forum about it. Bhavna, we'll close with, unless there are any new questions which pop up, a question from Nidhi Gujar, who asks, does LHX2 also impact the formation of layer 4?
So the answer is that we don't know. We have not removed LHX2 from the layer 4 to answer that. Maybe it does, it's expressed, so maybe it does, but the answer is that we don't know, and we need to check that.
Okay, and then there's a question asking, I think, about how you can control or regulate steps. I'll read the question out. Are there any pathways related to reverse processes that may restore functionality to the brain at any point?
So that's a very nice question. So it talks to that sort of developmental plasticity window which we have in the developing brain. So which means that so there are these studies where they overexpress FESF2 in these layer 4 neurons, and they could convert the layer 4 neurons into layer 5. Now there is a time window for doing this. It's not that you can now go into an adult and do this. So there is time window that you can do this at any time during development, and they've done this precisely at embryonic day, postnatal, just born pups. So to give the context, brain development happens even postnatally. So the first few years, and that's why also the brain is still developing, right? So just postnatally, if you overexpress FESF2, you convert those neurons 4 into 5. So this is possible. Again, this is sort of a reprogramming or something, and people are still trying to understand the mechanisms behind this. So in a window, it is possible to do that. Yeah.
Okay, great. Thank you very much, Bhavna. I think that's also a good note to close the talk on, because it sort of goes to the heart of what you were telling us. Thank you very much for your time this morning, and to all our audience, we'll see you again in 2 weeks. Okay. Thanks, Chandrika.
Thank you.